In this video, I wanted to just demonstrate how key flagging and note-taking are recorded into the save files. So we have two DRTs plugged in. They're called DRT 55 and 34. We're given that number by the software to keep them straight. We're going to create an experiment. We're now in the new folder. So we're going to select that folder. And we're going to collect some data. Well, let's write an optional name, a name. We're going to collect some data. Notice that as soon as we begin a block, we're no longer able to change the experiment name. Okay. Key flag here, I tried to change experiment name, but a key popped up here in key flag. This is important. So what happens is during a block, we're able to, and if we're not clicked inside the note box, if we're selected somewhere else, we're able to write a key flag. So in this case, A, B, C, D, whatever. And that flag, every time we press that button, that key is written to the output save file. The moment we press that button, a new key flag is added. Okay? So if, say, we're starting a condition that dynamically changes, and we have a participant, say, that's interacting with a device, and there's several steps to the interaction, we can flag each of those steps. So we can say step A, maybe the beginning, and then as soon as they move to the next step, we can say step B or C, or whatever that step is. And every time we press that button, that key is then written to the output file. Say a participant messes up, and we're like, oops. Participant failed to enter correct sequence or something. And we post that. Let's see what happens. Hit pause, end. We're going to come over here to our file, open last save location, open up our file, and here we have it. Okay, so to make this easier, we're going to paste this over into Excel. So you can see exactly how this works. Okay, so let's pay special attention to this key flag column here and this event or note column over here. Initially, when we began this experiment, I pressed a key, so we have a flag. I pressed an F and then an E. The times when I pressed F was recorded right here, down to the millisecond, with a little bit of actually microsecond precision there. Okay, and this is when I press the key E, down to the millisecond with microseconds. Okay, so the flag, the time when the flag is pressed is recorded in the output file. That becomes very important for synchronizing data and understanding under which condition the data were collected. So here I pressed E, and that flag then maintained its state through the next several DRT response trials that were read in. So when this DRT trial came in, when it was saved, it said what was the last key press that was down, and it saved that key press over here, that flag here in the key flag column. So we can know that then when this data came in, the condition E was present. So we can easily analyze then all of those DRT events that are associated with that condition based on the flag there. Okay. Later on, I started clicking some more flags. So we have several other flags that I pressed here, E again, S, A, and B. Each time every one of those flags were pressed, a new timestamp was created. And then when the DRT data came in, the currently pressed flag was associated then with that data. So we can see condition D for these DRT events, condition A for these events, C for these events. So this allows us to analyze precisely what events were associated with which DRT response times. There's an important caveat here, and that is this event here, any DRT event, when it comes in, it's assigned a flag. It's possible that when the probe was given to a participant, a different flag was pressed. And then we updated the flag after the probe was given, in which case when the data came in, the data would have been assigned that new probe, the new flag, and may not have matched the exact condition under which that response was collected. That's why it's important to consider the time in which the flag was pressed, the time in which the DRT stimulus was recorded, as well as the millisecond, this millisecond counter here of how long that trial had been going on. With all of these timestamps, it's possible then to figure out precisely the flag that was down when the DRT event was given. So there's some interesting timing things that need to be worked out there. But with a little bit of math and some careful coding, we can figure out exactly what flag was down when the DRT probe was presented, what flag was down when the response was made, etc. So because a DRT probe could occur over 3 to 5 seconds following the standard ISO procedure, there's a lot of potential time in which the flag can change. So just keep that in mind as you're analyzing the data. Down here we posted a note. Okay, so when a note is posted, here in the ID we get note. This is the time the note was posted, and this was the content of that note. And this was the flag that was pressed during that posting of the note. So we can keep notes in the same file as all of our data. So this is just an example of key flagging and note-taking with several Red Scientific devices plugged in and recording simultaneously. Again, if you have any questions, feel free to send me an email. I'm happy to help out any way I can, and we'll see you in the next video.